दिया आपको? Okay. So in yesterday's class, we left this enzyme portion of this topic. Today we will cover this, okay, in this class. Here, actually, in de novo pathway for purine biosynthesis, this pathway can be divided into two steps. The first step is synthesis of inosine monophosphate from basic precursors, and the second step is conversion of inosine monophosphate to AMP and GMP. So the first step actually here is catalyzed by the enzyme ribose 5-phosphate pyrophosphate phosphokinase. This ribose 5-phosphate pyrophosphokinase acts on the substrate ribose 5-phosphate and just transfer pyrophosphate group to carbon number 1, okay, from the ATP, okay, and ATP get converted into AMP, and we will get our first product of this pathway, that is 5-phosphoribosyl alpha pyrophosphate. This enzyme is the rate limiting enzyme, and this is also considered as a committed step of this pathway. So remember, this is very important. Okay, this PRPP via second step under the influence of PRPP amidotransferase is a transferase enzyme which just transfer the amino group from glutamine to this PRPP, and this PRPP now converted into phosphoribosyl beta amine. This phosphoribosyl beta amine again reacts with glycine and in the presence of ATP, the enzyme synthetase basically catalyzes the fusion of glycine to the phosphoribosyl beta amine, and it leads to the formation of glycinamide ribonucleotide. So this is our third step where we get the glycinamide ribonucleotide, and this glycinamide ribonucleotide further takes one carbon unit from N10 formyl tetrahydrofolate pathway in the presence of enzyme transformylase. What this enzyme does, it just transfer one carbon formyl group to this nitrogen of glycinamide ribonucleotide, and in this way this will become formyl glycinamide ribonucleotide, and this reactant in the presence of FGM synthetase, here the enzyme name is synthetase because it just transfer amino group from glutamine. We can also put here name the transferases, okay, because it just transfer the amino group of glutamine to this compound and lead to the formation of formyl glycinamidine ribonucleotide. And this formyl glycinamidine ribonucleotide in presence of enzyme synthetase, again one molecule of ATP required in this pathway, lead to the formation of next subsequent molecule, and which under the influence of our just let me put this bar somewhere else because it is not visible, I think. Okay, so it will lead to the formation of 5-amino imidazole ribonucleotide, and this in presence of carboxylase enzyme. Remember, carboxylase enzyme always incorporates CO2 to its substrate. Okay, lead to the incorporation of one CO2 group at carbon number 4, and there occur formation of carboxy amino imidazole ribonucleotide. In this step, it is actually directly written in here in the slide, but there is one step of ring closure where H2O get removed and your imidazole ring get formed. Therefore, it is converted into 5-amino imidazole ribonucleotide, and this then during subsequent reaction, this carboxy amino imidazole ribonucleotide in presence of synthetase enzyme just transfer aspartate, fuses aspartate to this COO group, and it will become N-succinylO 5-amino imidazole 4-carboxamide ribonucleotide. And this compound, in presence of lyase enzyme, whose full name is adenyl succinate lyase, fumarate get removed from this part and leaving behind the amino group, and it will become 5-amino imidazole 4-carboxamide ribonucleotide. And this, in presence of transformylase, again one carbon pool act here, and it will transfer the formyl group over this amino group, and there occur formation of N-formyl aminomidazole 4-carboxamide ribonucleotide. And this will, again in presence of synthase enzyme, there occur removal of water molecule and formation of inosine monophosphate. Okay, in this way, 11 step, this PRPP is completely converted into inosine monophosphate, and these are all the names of enzymes which you have seen at your left hand side, and you can memorize this. So here it is the first step of purine nucleotide biosynthesis, that is conversion of PRPP to the inosine monophosphate is completed. And now moving to the next step, this is just basic reaction. If somebody wants to memorize it, you can just see this, how one compound get fused to another, and the energetics of your purine biosynthesis. One should remember this because energy expenditure calculation in the body plays important role in overall physiological process. Here 6 ATPs are required in the purine biosynthesis, where ribose 5-phosphate is converted into inosine monophosphate. Since in one step ATP is directly, in the first committed step, ATP is directly converted to AMP, hence the total ATP requirement for de novo purine biosynthesis or for formation of inosine monophosphate only, IMP synthesis, is 7 ATP, okay, equivalent to 7 ATP. Now we will moving to the next step of the purine biosynthesis, that is conversion of IMP to AMP and GMP. And here in this step, basically aspartate, remember this fact, aspartate and GTP is used for AMP synthesis, and glutamine and ATP is used for GMP synthesis. Okay, here actually you can see this is very simplified picture for depicting the synthesis of AMP and GMP. Here you can see that the inosinate, that is inosine monophosphate, when it reacts with aspartate, enzyme is synthetase, there occur formation of adenylosuccinate, where this aspartate moiety is completely fused with this ketone group. Okay, you can just put, if you remember the structure of aspartate, that is COOCHNH2COO, you can directly put this structure over here. Okay, here one molecule of GTP is required as an energy source, and in the next step, the adenylosuccinate lyase enzyme is acting here, is carrying out this reaction, where fumarate get removed, and NH2 group is put at the place of 6th carbon, and now it becomes 6-aminopurine or adenosine monophosphate. So this is our this here in this way AMP get synthesized. In the second step, this IMP can also be utilized for the synthesis of GMP, where addition of water molecule and NAD, here enzyme is dehydrogenase actually, and it is IMP dehydrogenase, which basically catalyze the removal of this hydrogen and addition of oxygen at this group at carbon number 2, and there occur addition of first ketone group at the carbon number 2, and then this will lead to the synthesis of xanthylate, that is xanthine monophosphate from IMP. And this xanthine monophosphate, when it undergo the subsequent reaction in presence of transferases, the glutamine transfer its amino group at this ketone, and ATP is required as a source of energy here, and it will lead to the synthesis of guanylate. Okay, guanosine monophosphate. You can see here ATP is directly converted into AMP. That means it is equivalent to 2 ATP. So for the synthesis of GMP, 2 ATP molecule required, and glutamine is acting as a amino group donor. But for putting amino group at this position, 2 amino 4 oxo purine is our guanidine. For putting amino group at the second carbon, first this carbon should be carbonylated. That is here, this carbon should be connected with the oxygen in presence of a dehydrogenase. So you can see in this way, IMP is utilized for the synthesis of AMP and GMP. This is the overall reaction, how this AMP and GMP subsequently converted into ATP and GTP, because our deoxyribonucleic acid, that is DNA and RNA, is always having the basic units in the form of ATP and GTP, CTP and TTP. So this AMP in presence of kinase enzyme, the full name is AMP kinase, one molecule of ATP is needed in this reaction, is converted into ADP. Then again, subsequent reaction carried out by kinase, this ADP is converted into GTP. Similarly, GMP is converted into GDP and GDP is converted into GTP in presence of kinase and ATP molecules. So in this way, ATP and GTP get produced inside the body. How? Now coming to the next step, how purine nucleotide biosynthesis get regulated inside the body. So purine nucleotide biosynthesis is well regulated to meet the cellular demand. Actually, two enzymes are key regulatory enzymes for the purine nucleotide de novo biosynthesis. And the first enzyme, as I already told you, is PRPP synthase, which synthesize PRPP, phosphoribosyl phosphate. And actually, PRPP is a feed forward activator. Feed forward activator means we have learned about feedback inhibition. Okay, but this is something new. You can see feed forward means if PRPP is synthesized, it will activate the enzyme more rather than inhibition. Okay, so once PRPP gets synthesized, it activates the reaction in the forward direction or stimulate the reaction in the forward direction. So this PRPP basically, and this is also an activator of PRPP glutamyl aminotransferase enzyme. The intracellular concentration of PRPP regulates the purine biosynthesis, regulates the purine biosynthesis to large extent. Okay, to large extent. And the another factor is more the PRPP is available, more will be the synthesis of purine nucleotides. So it is actually, it is not inhibited by feedback control, it is activated by feed forward mechanism. IMP, AMP and GMP, if they are available in sufficient concentration, they basically inhibit the regulatory enzyme by feedback mechanism. So AMP and GMP, they actually undergo the mechanism of feedback inhibition. Okay. So PRPP basically activates by PRPP, or it also activates glutamyl aminotransferase, the next enzyme, and IMP, AMP and GMP inhibit the PRPP synthetase. If sufficient amount of AMP is present inside the cell, so it will inhibit the concentration, it will inhibit the second step, that is the conversion of IMP to AMP. And if sufficient amount of GMP is present, it will inhibit the conversion of IMP to GMP. Remember this fact too. And the regulation of in regulation of AMP synthesis, if we want to take some key notes, we should remember this enzyme, adenylosuccinate synthetase. It is actually feedbackly inhibited by AMP. So the first enzyme is PRPP synthetase and the second enzyme is adenylosuccinate synthetase. And IMP dehydrogenase, the enzyme which I talked you during the synthesis of GMP from IMP, this enzyme is feedbackly inhibited by GMP, IMP dehydrogenase. ATP stimulates the conversion of IMP to GMP and GTP stimulates the conversion of IMP to AMP. This ensures the balanced synthesis of both families of purine nucleotides. And what is the significance of regulation of de novo synthesis? Actually, why any pathway get regulated? Any pathway, the regulation of any pathway is very important just to meet the sufficient need of that compound, or in this case, nucleotides to body function without wasting. Otherwise, like nowadays, people takes more food in his or her plates and then they throw those food after eating leftover food, and that is very bad. But body is always working to balance each and every substrates just to avoid the wasting. Okay, so we should also follow this principle in our daily life. And here also AMP and GMP controls their respective synthesis from IMP biofeedback inhibition. So the balance between GTP and ATP actually maintained inside the body. So this is the significance of regulation of de novo synthesis of purine, and you can also see the significance of regulation of other pathway we have, which we were taught, learned in our earlier lectures. Okay, so purine nucleotide biosynthesis basically regulated by feedback inhibition, and in this way you can see you can see how each compound feedbackly inhibited its own key enzyme. Okay, like ribose 5-phosphate is converted into PRPP, and this PRPP basically feed forwardly activated the enzyme glutamine PRPP aminotransferase, and the availability of PRPP itself activated the first enzyme synthetase. But if there is sufficient amount of GMP and AMP in the cell, the GMP feedbackly inhibit the enzyme IMP dehydrogenase, AMP inhibit the adenylosuccinate synthetase, and if there is sufficient amount of GTP, it will inhibit aminotransferases and also this pyrophosphokinases. ADP similarly inhibit our aminotransferases and pyrophosphokinases. Similarly, if there is more amount of GTP and ATP present in the cell, GTP cross-reactively inhibit this adenylosuccinate synthetase, and ATP inhibited the IMP dehydrogenases. Remember, ATP and GTP just counterbalance their pathways so that their level should be maintained inside the cell. So in this way, we have just completed our regulatory part of this lecture, how the purine biosynthesis, de novo biosynthesis of purine regulated inside the body. Okay, now coming to the next very important part, which is important in perspective of competitive exams, that is antimetabolites or inhibitors of purine purine nucleotide biosynthesis. And this is actually, what is the significance of this antimetabolites or inhibitors? Their best significance, or we can tell advantage, is just to inhibit this pathway in certain crucial conditions when body get weakened by some disorders like cancer or antibacterial or bacterial infection. We can utilize this strategy to inhibit the synthesis of purine nucleotide just to stop the cancerous growth or bacterial growth inside our body. So this is very good pathway. Actually, all over world, people are rushing to just synthesize any new inhibitor to just stop nucleotide biosynthesis for bacterial or cancerous cells. Okay, and you can also see nowadays for COVID situation, people are just trying to synthesize best RDRP inhibitor, so that their nucleic acid replication get inhibited and uh, virus infection can be taken care of. Okay, so this is the significance and how these compounds works actually, how these compounds actually works. uh, antimet Antimetabolites of purine nucleotides are basically structural analogs of purines and amino acids and folic acids. Okay, so they can interfere, inhibit, or block the biosynthesis pathway of purine nucleotides, and further block the synthesis of DNA, RNA, and protein. This is widely used to just control the cancer as a chemotherapeutic agent. And the best known example of purine analog is 6-mercaptopurine, which is also known as 6-MP, is basically it is an analog of hypoxanthine. Okay. So this is the structure of 6-MP mercaptopurine, and here this is the structure of hypoxanthine, where at this position, at 6 carbon position, OH group is present and it is replaced by SH group, then it will convert it into 6-mercaptopurine. 6-mercaptopurine basically inhibit the adenyl succinase enzyme and AMP dehydrogenase enzyme, and it decreases the level of AMP and GMP in the cell. So this 6-mercaptopurine basically inhibit these enzymes, amidotransferases and HGPRT. If some of you studied earlier about monoclonal antibody synthesis, then you should be aware about this HGPRT, hypoxanthine